I know they're probably only off by a minute, but it always bothers me that that clock is a little different than what my watch says. Does everyone have 11:45 right now? Is it 11:45? No, what time is it? Okay. I'll wait another minute just in case. Oh, I set my watch back. That's fine. Sometimes I do that to avoid being late for things. But I always forget which clocks I did that to. Okay, so as you're getting set up, I threw a lot at you on Friday when we began this. I don't know if you've all noticed, but this course has a tendency to kind of speed up. It's actually supposed to throw even more at you on Friday, but I think we did plenty. But we're going to get started with some practice questions on that stuff on Friday. Okay. I'm going to get settled. Just trying to make sure that we're all on the same page, telling the difference between the different parts of our nervous system. If your memory's a little foggy, I would take a look at that very first slide. I'll take another couple seconds because I put a bunch of these in here. I don't want us to get too much more behind our schedule. Okay, let's take a look. Okay, great. So the answer should be C, afferent fibers. So those are our sensory fibers. So anytime you have information going from a receptor out in the peripheral nervous system to the central nervous system, so to either the spinal cord or the brain, that's always going to be part of your sensory nervous system, and we call those afferent fibers. So A, first it has to come in before it can go out as an efferent fiber. All right. The next thing we want to know is about those fibers that would be going out. Now, if we're transmitting information from the brain, and I'm going to say specifically to smooth muscle, think about what type of fiber that would be. Okay, a couple seconds, an answer, go forward. So, okay, yeah, so the answer should be F, that one where we have A, B, and C. So this is a little tricky. Both B and C are right, but so is A. So smooth muscle is one of our effector organs for the whole autonomic nervous system. So the autonomic nervous system, the whole thing, is part of the efferent branch. So all our autonomics, stuff going out. So autonomics are going to be one part, and somatics, when we get there, are going to be the other part. And autonomics all innervate pretty much the same effector organs. So that's smooth muscle, cardiac muscle, and glands. And so that means when we're thinking about any one of those things, we're going to expect to see both sympathetic and parasympathetics going to those same tissue types.
So you prefer to say that the afferent is to the brain, the efferent is from the brain?
Yeah, that's exactly right. Yeah. With the only tiny caveat that some loops will be a little smaller and only go to your spinal cord, which counts part of the central nervous system. So I think in phys, when you do reflexes, you measure one that just goes to the spinal cord.
So like instead of to the brain, just to like the central nervous system.
Yep. Okay, so now we're going to dive into those pieces of the autonomic nervous system. So if we're thinking about a neuron, specifically a preganglionic neuron, starting at T8, so that's a thoracic vertebra. Think about what part of the autonomic nervous system that would be. Okay, and then a couple seconds. Yeah, okay, so the answer is sympathetic. So the sympathetic division of the autonomic nervous system, specifically coming from the thoracolumbar region, is coming from the spinal cord in the area of your thoracic vertebrae, like your chest, and the first two lumbar vertebrae. When we think about the parasympathetics, they have to be coming from the brain. So we're going to see them on cranial nerves. You saw the cranial nerves in lab. Several of those carry parasympathetic innervation. And we'll also have a couple coming out of the pelvic region. They're not coming from anywhere in the middle. Okay. So if we think about a neuron starting at L2, now we're going to think about where it would synapse. So where it's going to link up with its postganglionic neuron. Okay, take another second or so. Okay, great. So most of you got this one right. So if we're starting in L2, that means this is a sympathetic neuron. Sympathetic neurons are going to synapse mostly in the sympathetic chain. So that's running along the spinal column in the thorax. They may also go out to collateral ganglia, which are just other ganglia, other little swellings out elsewhere in the body. Some of them are going to go all the way to the adrenal glands, specifically to the inside part of the adrenal glands, which is called the adrenal medulla. And they're going to synapse there with those chromatin cells, which are basically modified postganglionic neurons. So all three of those first options are describing possible routes for a sympathetic neuron. You don't really have either of the sympathetic or parasympathetics going to the adrenal cortex. If we were going all the way to the surface of an effector organ, we know that our parasympathetics tend to have much longer pre-ganglionic neurons. So they're going to have those ganglia much closer to where they're going. So that on the surface might be the case for a parasympathetic neuron. And when we left off, we were talking about our receptor types and the neurotransmitters that our different neurons are going to give off. And this is what we're going to pick up with today as well. So worth taking a look. Okay, another couple seconds. So the answer is actually this effector organ innervated by the sympathetics. When we talk about our receptors, our first sort of separation of different receptor types we're thinking about are our cholinergic receptors, which receive acetylcholine, and our adrenergic receptors, which receive norepinephrine and epinephrine. Okay, so the plates we see epinephrine and norepinephrine release are from our postganglionic sympathetic neurons. So the place we would find receptors for those is going to be on the organs that those neurotransmitters are going to. And where you might have gotten a little confused here is that on your pictures, certain neurons are labeled as either cholinergic or adrenergic. When we say a neuron is cholinergic or a neuron is adrenergic, that's talking about what it's releasing. But if we say a receptor is cholinergic or adrenergic, we're talking about what it's receiving. So if we're talking about, for example, the postganglionic sympathetic neurons, we call them adrenergic neurons because they release epinephrine or norepinephrine. but they are actually going to have cholinergic receptors on them where they start off because they're receiving acetylcholine from the preganglionic neuron. So we'll pick up with our type of cholinergic receptors. So the way I like to remember what's giving off acetylcholine versus what's giving off our adrenaline or norepinephrine, our epinephrine or norepinephrine, is basically your baseline should be thinking that something is releasing acetylcholine and that you should be thinking cholinergic receptors, because that's going to be true everywhere in our parasympathetic neurons. It's true of our preganglionic sympathetic neurons. We're going to see that muscles and the somatic parts of the nervous system also use acetylcholine. So the only place we're thinking about adrenergic receptors are those post-ganglionic sympathetic neurons and their effector organs. That's really important, and we will talk about adrenergic stuff a lot, especially as we move into the cardiovascular system. But it's easier to learn that that is the exception. And so if you don't remember, acetylcholine is a better guess. Our emphasis is often on adrenergic stuff, but that's because everything else can be considered kind of the same, so we don't focus on it as much. Okay. So when we're thinking about cholinergic receptors, there are actually two types. So they're slightly different in the way that they function. So nicotinic cholinergic receptor is basically just like those ionotropic receptors we saw in our previous unit. So we're just going to have our ions come in, we're going to have a cation channel open. This is going to cause depolarization. So our muscarinic cholinergic receptors are going to be more like those metabotropic receptors that we talked about. We're going to see a G protein when we talk about muscarinic cholinergic receptors. The effect is going to depend on the cell, because when we looked at G proteins, we saw that there's kind of that intermediary step, right, where we can have different types of secondary messengers in the cell. We're eventually going off to a different channel to let things through. So it's going to depend partially on the cell machinery, what type of response the cell has. So when we're thinking about the location of those cholinergic receptors, we see them in all of our autonomic postganglionic neurons. So at that synapse between preganglionic and postganglionic, regardless of whether we're talking parasympathetic or sympathetic, always going to be a cholinergic receptor, and specifically that's a nicotinic cholinergic receptor. You're going to see that. And conceptually, the way I like to think of that, right, is you're trying to send an action potential just into that postganglionic neuron. So it's easier to send action potential quickly, right? We're just trying to get down the sequence. So it makes a little more sense to have just that cation channel that opens. That may or may not be a helpful way to think about it for you, but that's how I like to think about it. So when we're thinking about our postganglionic fibers for the parasympathetic nervous system, we know they're also releasing acetylcholine, but on those effector organs where their targets are, we're going to have muscarinic cholinergic receptors so that we can have kind of a varied response in those target cells in those effector organs. We may want to change it depending on what type of tissue we're talking about. Skeletal muscle, when we get to the somatic nervous system, which is what we're doing next, always nicotinic cholinergic receptors. Our muscles are basically sending off their own action potentials to contract. So that's why I think of it that way. The exception to all of this stuff is that our sympathetic effector organs pretty much always have adrenergic receptors, except sweat glands. So sweat glands are the only part of the effector organs for the sympathetic nervous system where you would see a cholinergic receptor. Specifically, they'd be muscarinic cholinergic. And the way that that happens is basically, you know, that the you don't know. The adrenal glands are releasing those hormones, the norepinephrine and epinephrine. So sweat glands can respond to like circulating stuff in the blood, basically. Okay. So our cholinergic receptors only came in 2 types, nicotinic and muscarinic, but our adrenergic receptors have 5 types. They're in 2 main classes to separate them out. So if you're trying to figure out if a receptor, which type it is when we're thinking about adrenergic receptors, you first want to figure out if you're thinking about an alpha receptor or a beta receptor. So they're all going to be coupled to G proteins, but the division is really going to be coming down to their interaction with cyclic A&P. So we're going to start with the alphas. So the alpha adrenergic receptors, like all of our adrenergic receptors that we're thinking about, are going to be in effector organs for the sympathetic nervous system, cardiac muscle, smooth muscle, and glands. Alpha adrenergic receptors are the more common type of receptor for our sympathetic nervous system, and they're usually excitatory, meaning they're usually creating that depolarization in this effector organ cell. They have a greater affinity for norepinephrine than epinephrine. Right, so here we see a picture of specifically an alpha-1 receptor. So it has a G protein like all our adrenergic receptors do. And G protein is triggering this IP3 molecule, which is then creating a response in the cell. Now, we're going to differentiate those alpha receptors from our beta adrenergic receptors. The beta adrenergic receptors all activate cyclic A&P. So I'm emphasizing the word activate here because in this image, we actually see an alpha-2 receptor on the left and a beta receptor on the right. And we can see that they're both feeding into the same system, the cyclic A&P system. But what the difference is here, you have to look quite closely. This is not necessarily super fair of this diagram, but you'll notice on this alpha-2 side, we have a minus sign. So that's saying that the alpha-2 receptor is inhibiting the cyclic A&P. So even though it's interacting with this second messenger system, because it's inhibiting or causing us to create less cyclic A&P, that makes it an alpha receptor. So all our beta receptors are going to be activating that system. So there would always be a plus if it's a beta receptor. Something that is going to vary between our beta adrenergic receptors, their affinities for norepinephrine and epinephrine, a little bit of a difference in their location, and a little bit of a difference in whether they're excitatory or inhibitory. I'll see one exception there. So for our beta 1 adrenergic receptors, we're going to find them in the heart, we're going to find them in the kidneys. They're going to be excitatory, so they're going to cause depolarization. And they have they have an equal affinity for norepinephrine and epinephrine. So that receptor, that protein, doesn't really care which neurotransmitter is coming in. They both fit equally well into their active site. The beta 2 adrenergic receptors are inhibitory. So that would be one of our big clues that you're looking at a beta 2 receptor. We're going to find them in some blood vessels and some smooth muscle, much of which surrounds blood vessels. We'll also see smooth muscle in the digestive system. Beta 2 receptors, the other thing we're going to notice about them is they like epinephrine much better than norepinephrine. So they do actually care which neurotransmitter they're receiving. Our beta 3 adrenergic receptors are usually located in adipose tissue, so in fat. Like the beta 1 receptor, they're usually excitatory, and like the beta 1 receptors, they have equal affinity for norepinephrine and epinephrine. So, quick question, describe a receptor. I think you have a table in your slide, so you could compare the table as you're answering this question. Thinking about a receptor that has high affinity for epinephrine, creates an inhibitory effect in some blood vessels using cyclic A&P. What type of receptor would this be? To parse this question out. So one thing you're paying attention to, that we're thinking about this neurotransmitter. The second sentence might lead you a little bit astray. So I'm going to say that we're activating cyclic A&P, but the effect on the cell, on the blood vessel, is inhibitory. And that should get you where you're going. Great, yeah, so that's that beta 2 receptor. So this first part, because we see the word epinephrine, we know we're talking about an adrenergic receptor, not a cholinergic receptor. Since I told you out loud, and I should have written it in the question, activating cyclic A&P, that makes it a beta receptor. And then because we are inhibiting the blood vessels, that means it's a beta 2 because the beta 1 and beta 3 are excitatory. The other way we could tell there is the beta 1 and beta 3 have equal affinity for epinephrine and norepinephrine, but that beta 2 receptor prefers epinephrine. So as you work through practice questions like this, or as you create yourself and your friends practice questions, consult the table. Right, so we have effector organs that we're thinking about, or the receptor types that we'd expect to see in those places, a chart of which neurotransmitter they prefer. The signal transduction mechanism is telling us how it interacts with the cyclic A&P system, or if it doesn't in the case of the alpha 1 receptor, and then whether we have an excitatory or inhibitory effect. And this is just for your interest for the future. So part of the reason we care about all these receptor types is that a lot of different drugs target these receptors as part of the way that they work. So you can see here that these first two receptors are agonists of the muscarinic receptors. So basically they like upregulate them. We can see that we have alpha agonists and antagonists, so like the antagonists fight them, and same deal with these beta 1 receptors. So this is a useful place to think about drug targets. We're going to look now in detail at that linkage between our postganglionic autonomic neuron and the effector organ that they are affecting. So we're going to see a type of synapse here. We're going to call it a neuroeffector junction. So neuro for that postganglionic neuron, effector for the effector organ. And it's going to look pretty similar to how we saw our synapses working in the brain and in the central nervous system, with just a couple of differences. So the main difference that we're going to see is that rather than coming from an axon terminal, we have these special structures called varicosities in our postganglionic neuron. And like basically the end of a postganglionic neuron looks like a hand with these like weird spaghetti fingers spreading over the affected organ. So it's this kind of diffuse spread out effect. It's not targeted in just one little place. So each of these swellings is something called a varicosity. And so each one of those swellings is going to work pretty basically like our axon terminal did in the central nervous system. So in each of these varicosities, we're going to be releasing neurotransmitter to that effector organ. So that means we could list out the steps here. So just like in our central nervous system, we start with our action potential coming down the axon. And now we're reaching a varicosity. So here we see acetylcholine from a postganglionic neuron. So would this be sympathetic or parasympathetic? Maybe contrast. This other one is going to be the opposite. So one of these is sympathetic and one of them is parasympathetic. So this is the parasympathetic because that's a postganglionic releasing acetylcholine. And here norepinephrine means this is a sympathetic postganglionic neuron. So there are neuroeffector junctions for both of these. Our action potential is coming down, opens those voltage gated calcium channels, just like we saw in our synapses in the brain. That calcium triggers the release of our neurotransmitter from the vesicles. So then that neurotransmitter is crossing over the space. So before we called this place a synaptic cleft. Now it's not a really discrete cleft. There's a bit more space here. They're a bit farther apart, but the same type of thing is happening. We have this neurotransmitter crossing and binding on to a receptor, now on our effector organ rather than on another neuron. There's a response of some type, and then the neurotransmitter either diffuses away, gets broken down, or is taken back up into the varicosity. So that all works exactly the same between the parasympathetic and sympathetic neurons. The only difference is which neurotransmitter they're releasing. Here's some questions for you to consider later. We're going to skip through those right now, and we're going to think about that dual innervation part of the autonomic nervous system. So that question at the beginning, where I forget exactly what the question was, but where we said that we had that something was part of the efferent nervous system and could be parasympathetic or sympathetic. This is because we have the dual innervation of organs. So an organ, say your heart, receives both sympathetics and parasympathetics. It's not like the heart gets sympathetics, but your lung gets parasympathetics. They both get both. So there's always a balance. So we're thinking about like a dimmer switch between whether there's more parasympathetic or more sympathetic, but we're not really going to be at zero for either. The parasympathetics dominate at rest, and the sympathetics dominate in sort of stressful situations, that fight or flight situation. So we think about them as involved with excitation, basically, right? Making your systems more active. So if we increase the activity of the parasympathetic nervous system, the sympathetic nervous system usually decreases its effects at the same time, and vice versa. So if we increase sympathetic activity, we're going to be decreasing parasympathetic activity. So we have an example here of an autonomic reflex response. So something that involves both the parasympathetics and sympathetics and their balance. So when you stand up, it is true that gravity actually pulls on your blood. So when you stand up, gravity is pulling your blood down away from your head and causing it to pool in the lower limbs. This means that in your arteries and the rest of the body, you're actually seeing a drop in blood pressure. So this is one of the reasons why if you've ever stood up and suddenly your vision goes like gray and starry, that's what's happening actually. It's that change in blood pressure. So we have a reflex to get your blood pressure back up to normal, make sure your brain's getting the blood it needs. So we have receptors out in the body that detect that decrease in blood pressure. So when we get to the cardiovascular system, we'll talk about those receptors. But the way that they signal to the brain, to the central nervous system, that we've had this fall in blood pressure, is that they are basically the receptors always firing action potentials to the brain. And if those action potentials slow down, the brain knows that your blood pressure has dropped. So this is an afferent pathway because this is like a sensory receptor sensing how high your blood pressure is. So when we find that signal coming into the brain, you don't need to know this, but specifically that control center is located in the medulla oblongata that you saw in lab. And so that's where we're going to create this response, this reflex response, maintaining homeostasis. Blood pressure is something we want to control because having enough blood to the brain is super important. So the way we get blood pressure back up is we increase sympathetic activity, which acts on the heart and blood vessels in such a way to increase the blood pressure. And we'll talk about that in great detail in the future, but it basically has to do with changing diameters of blood vessels and making your heart beat harder.
Why does the blood pressure affect an action potential?
So that's basically how the receptor works. It's saying that the afferent nerve, like the way it works is that you have this constant flashing light, right? This constant action potential. And if it changes in speed, like if it slows down, or if it were to speed up, that's what the signal is to your brain, right? Like an afferent neuron can't send, right, like a little note that says like, hey, the blood pressure has changed. It all has to be signaled in terms of action potentials, like, because that's the only thing a neuron can do is change action potentials. So you might think initially that like the neuron is just dead and not firing action potentials when we're at normal, but that's just not the case. It's that it has a constant rate that the brain expects, and the way the brain knows something has changed is with the rate change.
So if the blood pressure drops, then action potentials don't.
They're
Like,
slowing down.
Okay, so I guess like I don't understand how like the pressure in the blood vessels like.
Yeah,
big.
it's something about how the receptor works. That's just how the receptor functions. Yeah. We'll talk about receptors in the future, although honestly, we won't get super into detail about how they all exactly work on that level. But basically the cellular mechanics inside the cell have this effect of changing the frequency of the action potentials. So like those in specific are baroreceptors, so they sense changes in pressure. So basically kind of stretch in the walls of these arteries causes the change in the ion channels, basically. But we have other types of receptors that sense like different chemical concentrations and stuff like that. So it would depend on what type of receptor. Okay, so yeah, so when the action potentials drop, the brain knows that the blood pressure dropped. So to get it back up, we increase sympathetic activity. We don't totally turn off the parasympathetic activity, we just turn it down a little. This increases the blood pressure, which means we then have a negative feedback loop. So now that your blood pressure is back up, the blood isn't pooling anymore. So the blood pressure went back up, right? Which means that we're no longer going to sense this decrease in blood pressure. So the frequency of action potentials would go back up to where it normally is, which means that the integration sensor could turn on the parasympathetic activity a little more and turn down the sympathetic activity just to kind of keep it back where it was. So a basic understanding of the parasympathetics and the sympathetics, that our parasympathetics are active in our rest and digest states. And this means that they're going to be increasing the activity of your GI system. So that's the digest part. So the parasympathetics are going to be triggering yourself to like take in glucose, things like that. may decrease heart rate and blood pressure because you wouldn't expect, like, say, your skeletal muscle to need tons and tons of blood. You're not running away from anything. You're just kind of sitting having a snack. Whereas the sympathetic activity is trying to prep you to either run away for something or fight it, which means that you're going to need a lot of blood to your muscles to do stuff. And you're going to need to breathe a bunch, right, to get oxygen. So you're going to be increasing the heart rate, increasing blood pressure to get that blood and oxygen quickly to your muscles. We're going to start mobilizing energy stores. So we're going to be pulling glucose out of our cells, out of glycogen, in order to make ATP in the muscles. It's true that at the same time it dilates your pupil. It's going to decrease your GI functions and urinary functions because you don't really want to stop and pee or something as you're running away from something. So we refer partially to this dual innervation system where both are on, but just in differing levels as tonic activity. And at rest, our parasympathetic nervous system is dominating. I am not actually going to question you about this, but these are the regions of the brain that are particularly involved with autonomic functions. So you've hopefully seen some of these in lab. We have our hypothalamus. We're going to talk a lot about as we talk about our endocrine system as well. We have parts of the pons, which we have respiratory sensors in there. We have cardiovascular sensors in there as well on the more posterior part. And the medulla oblongata also is involved in this function. So those are the locations where we'd expect to see those integration centers. So mostly in the brainstem. Okay. And here are the exact effects. I've been using the heart a lot as an example, but we do have effects all over the body of different types and that are involved with different adrenergic receptors, specifically in the sympathetic nervous system. The parasympathetics are really just thinking muscarinic cholinergic receptors. We'll talk at length about the effects on like blood vessels and lungs as we move into those units in the future. So here's just a quick cheat sheet for that. This may be helpful to come back to as we get to those organ systems. So we're going to be contrasting the autonomic nervous system with the somatic nervous system. You'll notice that your packet for this is only like 12 sides long or something. The somatic nervous system is hopefully pretty simple. So we're going to look at the anatomy of the somatic nervous system, basically just how their form of synapse, the neuromuscular junction, differs from a neuroeffector junction. And we're going to take a look at a couple of toxins that affect this, just to kind of understand that junction a little better. Okay. So you may have seen this already in phys lab, but the somatic nervous system, the part of the efferent nervous system, is innervating skeletal muscle. So motor neurons going to those named muscles attached to the skeleton. That's the voluntary part of your efferent division of the nervous system. Specifically, they're coming from the ventral horn of the spinal cord here when we have those neurons coming out. They're going all the way out to the skeletal muscle. This is a much simpler route than when we're thinking about the autonomics. We don't have to think about differences between preganglionic, postganglionic. We just have this neuron going to the muscle. The neurotransmitter these neurons are releasing is acetylcholine. The receptor in the skeletal muscle is a nicotinic cholinergic receptor. A thing that we will want to know about muscles, and we will touch back on this as we get to our muscle physiology and activity at the end of this unit, so that a motor unit are all the different muscle fibers that are innervated by a single motor neuron. So motor neurons split and send signals to multiple fibers, but not like every fiber in your biceps is innervated by one neuron. So you have multiple motor neurons going to a named muscle, but they have a group of fibers within that muscle that they're activating. Oh, yeah, and just this last piece, this is not true if you're thinking from the muscle fiber side. A muscle fiber does only have one motor neuron that's controlling it. So we don't have dual innervation of motor fibers here. So if we think about the neuromuscular junction, so the point at which we're synapsing between our neuron and our muscle, like in our central nervous system, whether that neuron fires has to do with the summation of the signals coming in. So our EPSPs and IPSPs have to add up together to get us to that -55 to send off an action potential in the neuron, just like in our other neurons. Then eventually at the end of that axon, we're going to release our neurotransmitter acetylcholine. So what's going to happen after that is that on the other side of this neuromuscular junction, we're going to create what we call an end plate potential at a motor end plate. So that's part of this neuromuscular junction. And we have a slide of key terms. So now that we're looking on the muscle side, so we have our neurotransmitter coming in. So in this case, we're thinking about our neurotransmitter as being acetylcholine, right? So we're forming a synapse between our motor neuron and our muscle fiber. So specialized terminology for things that we basically already know the structure of. When we're thinking about the neuromuscular junction, we use the word terminal bouton for the axon terminal. Just has its special name if we're thinking about a motor neuron. And the motor end plate is the muscle membrane on the other side. So like this would be our motor end plate, right, where we see the receptors. So all our motor neurons are releasing acetylcholine, and on the motor end plate, we're going to see nicotinic cholinergic receptors. They're always going to be excitatory. So we're always going to create a depolarization, which we're going to call an end plate potential here in the cell, in the motor cell. Acetylcholinesterase is the name of the enzyme that clears the acetylcholine out of this space in the neuromuscular junction. And like for our other synapses, we have this numbered out numerically. So our steps here, we have our action potential coming in to our terminal bouton. Like before, we're opening up voltage-gated calcium channels. So we have calcium flowing in, triggering the release of these synaptic vesicles that hold acetylcholine. So the acetylcholine is getting released into this space. And the acetylcholine is binding on to that cholinergic receptor, that nicotinic cholinergic receptor. That's something that we want to remember for our specific receptor type in the neuromuscular junction. And now we have the net movement of positive charges in. So as we can see, that's our friend sodium again. We got a bunch of sodium come in, causing depolarization, which here we're calling our end plate potential. Special name because it's in a muscle, but it basically works like we saw in our other locations. That end plate potential causes an action potential in the muscle cell, which is going to eventually cause the muscle cell to contract. And the acetylcholinesterase is going to break down and clear out some of that acetylcholine. And the choline part of the acetylcholine is going to get sucked back into the cell. So we call that reuptake. We'll have reuptake happening here of the choline. We're not going to watch this little video, but if you want to hear the robot show you the neuromuscular junction, you have the link there. But what I did want us to get to was the toxin. So we'll do this real quick. So this is just to kind of think about how we might change the effects of this neuromuscular junction and what kind of response that's going to create in our muscle cells. So one type of toxin called rhodopsin, it stimulates acetylcholine release. So what that means is we're going to be releasing more acetylcholine. So more acetylcholine is going to be coming into this cleft. So what kind of effect do you think that's going to have on our muscle? What would a lot of neurotransmitter here do? Yeah, exactly. So we're going to, action here. So we're opening a lot of these channel receptors, right? And that means that we're going to be contracting that muscle a lot. So the muscle, in response to all this extra acetylcholine, is going to spasm. And at a certain point, it's going to become rigid, right? Because it's going to be like basically all contracted and kind of stuck there. We can contrast this with a toxin called protoxin, which inhibits acetylcholine release. So what do you think would happen in the muscle if we inhibit acetylcholine? We have less neurotransmitter. Yeah, exactly. So it experiences something we call flaccid paralysis, but basically that just means it's all loose and you can't contract it, right? Paralysis is not being able to use your muscles, right? So that's what's happening here. And then we have curare, which is in poison dart frog. What this does is block our nicotinic cholinergic receptors. So what do you think is going to happen here? Which is this going to be a little more similar to? Is it going to be more similar to inhibiting or stimulating acetylcholine release if we block a receptor? Yeah, exactly. It's going to be kind of like inhibiting those receptors. So this is actually also part of what's in muscle relaxants. So to make your muscles kind of calm down and relax, we can block those receptors, right? And if we block a receptor, even when we have neurotransmitter, it can't get in. So we have that same kind of loosening effect. Okay, so we will pick off with the endocrine system next time on Friday.